Welcome back to the deep dive. Today we are diving into your second brain, your gut bacteria.
It's a topic that for years was just, well, it was overlooked.
Completely. I mean, for the longest time, the scientific community, and let's be honest, all of us, we treated our digestive system like a really efficient waste processing plant.
Right, it was just a waste station. Food goes in, waste comes out. End of story.
But the science is finally giving us this crystal clear picture that the incredible complex ecosystem living inside us doesn't just digest food. It pulls the strings on our mood, our weight, our immune system.
Pretty much every other system in our body, yeah. It's a paradigm shift.
Okay, let's unpack this with a great analogy, because the idea of trillions of microscopic residents influencing your entire body, from your toes to your brain, is genuinely mind-blowing.
It really is.
And we're going to need to explain these incredibly complex medical meta-analysis results in the simplest possible terms, like we're talking to a very smart 8th grader.
Yeah.
So think of your gut not as a simple tube, but as a massive, bustling city.
Or, and I love this one, a lush, dense rainforest ecosystem.
Yes, a rainforest. That's perfect.
That analogy is perfect because it immediately captures the sheer complexity and the density of it all. This city is home to trillions of inhabitants.
Trillions.
Bacteria, yes, but also fungi, and even viruses, which we call bacteriophages or just phages. This whole community weighs about 3 to 4 pounds.
So it's like an actual organ.
It basically is. Its total genetic information is about 150 times greater than your own human genome. It's a massive hidden organ.
Wow. So we're carrying around a hidden organ made entirely of tiny organisms. And the rule of this city is simple, right? When the ecosystem is healthy and diverse, meaning all the different types of microbes are balanced and thriving, the whole body thrives.
Precisely. The city is productive, the infrastructure works, all the supply chains are running smoothly.
But the moment that microbial city falls into disarray,
Which scientists call dysbiosis.
right, dysbiosis, that's when the troubles start. And here's the key insight we're following today. Those troubles don't just stay localized in the gut.
Not at all. They manifest far, far away up in your brain, causing mood disruption, in your bloodstream, causing inflammation, or in your energy factories, leading to metabolic issues.
So we've got a problem of widespread systemic failure rooted in this tiny city inside us.
And our mission in this deep dive is to explore one of the most exciting and perhaps, controversial therapeutic approaches aimed at restoring order to that struggling microbial metropolis.
Fecal microbiota transplantation, FMT.
That's the one. We are taking the data from the most rigorous recent meta-analyses and systematic reviews, basically the scientific consensus, to gauge its true power and, just as importantly, its limitations.
And we're not talking about minor issues. We're talking about FMT being used to treat major complex diseases like chronic depression and widespread metabolic disorders.
Exactly.
For the listener who might have just heard the term, it sounds extreme, but the core concept is surprisingly simple when you think about it.
It really is. Think of it like this. If your struggling city is suffering from a lack of key workers, or maybe the sanitation department has completely failed.
The whole system is breaking down.
Right. FMT is like moving an entire healthy community, a thriving set of beneficial residents, from a donor's robust city into your struggling one.
So you're basically calling in reinforcements.
The ultimate reinforcements. The goal is that these new productive residents will set up shop, restore the supply lines, and bring the ecosystem back into balance. It's a complete microbe transplant.
And based on the research, there are 2 major highways we need to explore today.
That's right. We're going to focus first on the gut-brain axis, exploring that deep connection between microbial balance and the mind, specifically how a microbe transplant might improve conditions like depression. And second, we'll look at the gut metabolism axis, analyzing the surprisingly complex relationship between the gut and our ability to manage weight, control blood sugar, and fight that systemic inflammation we were just talking about.
So let's get into the mechanics. This is the essential how question.
Yeah.
Right? For the listener trying to grasp the magnitude of this connection, how can tiny bacteria sitting in my digestive system possibly influence something as massive and complex as my mood or my weight? It truly feels like science fiction.
It does, but it's not magic. It is, however, a monumental communication feat. The connection is what we call the microbiota-gut-brain axis, or the MGB axis.
Okay, the MGB axis. Let's simplify that.
Let's call it a secret superhighway, or maybe a sophisticated, highly efficient two-way telegram line. This line connects the deepest part of your intestinal tract, where the microbial city lives, directly to the command center in your brain.
A two-way street, constantly communicating. So what are the lanes on this superhighway? What's carrying the messages?
There are three main channels for this flow of information. First, you have the physical nerves, primarily the mighty vagus nerve.
The vagus nerve, I've heard of that.
It runs directly from the brainstem down to the abdomen, basically acting like a high-speed fiber optic cable. Second, the immune system, the body's internal police force, is heavily influenced by the gut. But most importantly for our discussion today,
The third channel.
the communication happens through chemical products that the microbes manufacture and then release into the blood, where they act just like hormones.
So the bacteria aren't sending emails, they're sending molecules, actual chemicals that act as commands and requests to our bodies.
Exactly. This is why we call the gut the second brain. The microbes are constantly manufacturing chemicals that the host body uses, sometimes for good and sometimes for ill.
Okay, so let's talk about the currency of this internal economy. What are the specific chemical messages these microbes are producing that influence everything from appetite to inflammation?
The primary form of currency is a group of essential compounds called short-chain fatty acids, or SCFAs.
SCFAs.
Right.
These are produced when our gut microbes do something we can't. They eat the fiber, the non-digestible carbohydrates that we get from vegetables, fruits, and grains.
So the stuff our own bodies can't break down.
The microbes ferment this fiber, kind of like brewing beer, and the byproducts, the SCFAs, are spit out. The major ones are acetate, butyrate, and propionate.
So if we eat a tough piece of broccoli that we can't break down, the bacteria handle it, and the reward for that work is this essential SCFA fuel for the rest of the body.
Precisely. And this fuel is vital. SCFAs are the essential preferred fuel for the cells lining the intestinal tract itself. They keep those city walls strong and healthy.
Without them, the walls get weak.
Exactly. If those cells aren't properly fueled, the walls start to crumble. But the SCFAs don't just stay local. They also travel through the blood to distant organs.
How do they talk to the immune system? You mentioned that was one of the channels. They act as diplomatic messengers, they travel through the bloodstream, and they talk to your immune cells by latching onto special receptors on the surface of those cells. Think of these receptors like GPR43 or GPR109A as very specific locks.
And the SCFA is the key.
The SCFA is the key. When it fits into the lock, it sends a message to the immune cell essentially saying, everything is under control, calm down. This is how SCFAs modulate the immune response and keep inflammation from running wild.
That makes perfect sense for gut health. But I understand these SCFAs have a fascinating dual nature when it comes to weight and metabolism.
Yeah.
They can be great, but they can also be part of the problem depending on the state of the city.
That's the critical nuance the research has uncovered. On the one hand, on the beneficial side, SCFAs are amazing at metabolic control. They promote appetite suppression, making you feel full faster.
Oh, wow.
They help the body burn stored fat for energy, a process called fat oxidation, and they aid in general energy expenditure. They even encourage the growth of brown fat, which is the good fat tissue that helps you burn calories just to stay warm.
They sound like the perfect employees.
Yeah.
So where does the trouble start? How do they turn bad?
The trouble starts in the context of an already unhealthy gut, that state of dysbiosis we mentioned. If the microbial city is overpopulated by the wrong kinds of bacteria, they can produce SCFAs, particularly acetate, in excessive amounts.
A flood of them.
A massive flood. And when the liver receives this huge flood of acetate, it can misinterpret the signal and use that excess to synthesize fat, contributing to lipid accumulation and increasing the amount of energy the body extracts from food.
So the same chemical currency that usually fuels healthy tissue can, in a dysfunctional system, lead to the hoarding of fat.
Exactly. And this is often seen in individuals who are already insulin resistant. Their body's hormonal and signaling systems are already altered, meaning the positive effects of SCFAs we see in healthy subjects might just not apply. The state of the microbial city determines whether that currency is used for construction or for overstorage.
Beyond fuel and inflammation, these microbes also influence the actual building blocks of the brain's mood chemistry, right? This is the link to depression.
Absolutely. They process key building blocks, most famously the amino acid tryptophan.
The Thanksgiving turkey chemical.
The very same. Most people know tryptophan as the compound that makes them sleepy after a big meal, but it's a crucial precursor for brain function. Microbes can convert tryptophan into two major and competing families of compounds.
Okay, tell us about the two paths tryptophan can take.
The first, The widely known path leads to serotonin, or 5-HT. Up to 90% of the body's serotonin is found in the gut.
90%. I thought it was a brain chemical.
Mostly made in the gut. Serotonin profoundly impacts mood. It's the classic feel-good neurotransmitter, but it also helps regulate gut motility. If your microbial citizens are healthy, they help ensure tryptophan moves down this mood-boosting pathway.
And the second path is the one linked to trouble.
Yes. The other path creates kynurin metabolites. These compounds are deeply involved in regulating immune responses and inflammation throughout the body. When there is chronic inflammation, that smoldering fire we talked about, more tryptophan gets shunted down this inflammatory kynurenine pathway.
Which means there's less available to produce serotonin.
Exactly. It's stolen for the war effort, so to speak.
So we have a clear chain of events. A miserable, dysbiotic microbial city creates inflammatory messengers, which then actively disrupt the brain's ability to create mood-balancing chemicals like serotonin because the tryptophan is being rerouted for inflammatory defense.
That is the core hypothesis of the gut-brain axis. And on top of that, the microbes influence levels of GABA, which is like the brain's main brake or calming neurotransmitter, and phenylalanine, another important amino acid. The chemical activity inside your gut is directly linked to the signaling pathways in your brain, governing whether you feel calm or anxious, motivated or depressed.
This is a perfect point to transition to the leaky gut idea. We talked about the city walls. What exactly does a compromised intestinal barrier mean, and why is that so fundamentally damaging to the rest of the body?
The wall of your intestine is made up of tightly packed cells held together by strong seals, like high-security gates. It's designed to be highly selective, only allowing fully digested nutrients through.
So it's a very tight border.
It should be. In dysbiosis, those seals, those cellular junctions, can loosen. This increases intestinal permeability, which is the technical term for leaky gut. When the city walls are damaged, they allow inflammatory compounds to slip right out into the bloodstream, where they absolutely should not be.
And when those unwanted compounds escape, they travel everywhere, triggering alarms. Which specific compounds are the main bad actors that cause this systemic havoc?
The most dangerous bad actor is a molecule called LPS, or lipopolysaccharide.
LPS.
Think of LPS as the tiny, spiky, toxic component embedded in the outer wall of certain types of bacteria, specifically gram-negative bacteria. These types are often dramatically increased in conditions like obesity or metabolic syndrome. When they die, or when the gut wall is leaky, this LPS slips out.
What happens when it hits the bloodstream?
Even a low-grade elevation of plasma LPS, a phenomenon we call metabolic endotoxemia, is enough to trigger a massive alarm signal across the entire body. The LPS travels to immune cells and docks onto a specific receptor, the TLR4 receptor.
Let's simplify that. What is TLR4?
TLR4 is essentially the main danger intruder,
Right.
alarm bell on your immune cells.
A red button.
Exactly. When LPS hits it, it activates a destructive chain reaction inside the cell, which we call the NF-key signaling pathway. This pathway essentially turns on the genes that produce massive amounts of inflammatory chemicals.
So a broken gut wall leads to an escaped bacterial component, the LPS, which hits the immune cell alarm, TLR4, and starts a low-grade chronic fire inflammation throughout the entire body.
Precisely. This silent, persistent inflammation is a major driver of modern metabolic diseases. It leads directly to insulin resistance, making your cells stop responding to insulin, and drives conditions like non-alcoholic fatty liver disease. It is the persistent, low-level smoldering fire that the gut barrier malfunction allows to ignite throughout the entire system.
That detailed mechanism makes the stakes very clear. If the gut is literally driving systemic inflammation and hijacking our mood chemistry, then fixing the gut should, in theory, offer a powerful new way to treat these complex disorders. Let's start with mental health, specifically depression. This is a staggering global public health challenge.
It absolutely is. We have to acknowledge the scale of this problem before we dive into the data. The World Health Organization confirmed that over 280 million people worldwide are affected. And tragically, depressive disorders have been ranked as the second leading cause of years lived with disability globally for nearly a decade.
And for a significant fraction of these people, the standard treatments, whether it's medication or psychotherapy, they just don't provide adequate relief.
Not for everyone, no.
Which is why researchers are desperately exploring these outside-the-box therapeutic strategies like FMT. So what did the recent massive meta-analysis of fecal microbiota transplantation show regarding reducing depressive symptoms?
The findings were surprisingly robust and highly compelling. We looked at a major meta-analysis that synthesized data from 12 randomized controlled trials involving over 680 participants. It found that FMT significantly reduced depressive symptoms overall.
Okay, let's put that in perspective for the listener.
Yeah.
They use this statistical measure, right? The standardized mean difference, or SMD.
Right.
The number was minus 1.21.
Yeah.
What does an SMD of minus 1.21 actually mean in terms of impact? Is that good?
It's fantastic. Think of SMD as a way to standardize the effect size across different studies so we can compare apples to apples. An effect size of minus 1.21 is considered a very large effect.
How large?
To offer some context, major pharmaceutical interventions for depression often target an SMD of around minus 0.5 to minus 0.8. So negative 1.21 suggests that the microbial intervention, on average, provided a therapeutic impact that was statistically and clinically powerful.
That suggests a powerful effect, almost equivalent to a major breakthrough drug, if we're just looking at the numbers.
Exactly. And the finding was highly resilient. They did what's called a sensitivity analysis, where the researchers intentionally tried to break the finding by removing studies that might be skewing the results. And the effect remains statistically significant and robust.
So it's a solid result.
It strongly supports FMT as an effective adjunctive therapy, meaning it helps significantly when used alongside standard psychological treatments.
But as you warned, the benefit wasn't evenly distributed across all patients. And this detail is key to understanding who this treatment is for.
This is the most critical piece of mechanistic evidence. The positive effect of FMT was notably stronger, dramatically stronger, in patients who also had a diagnosed gastrointestinal condition, specifically irritable bowel syndrome or IBS.
Ah, so the people with gut problems got the most benefit.
By a long shot. For these IBS patients, the reduction in depressive symptoms was much larger, an SMD of minus 1.06. You compare that to patients who were enrolled solely for neurological or psychiatric conditions without that strong gut component.
And the effect was smaller.
Noticeably smaller, yes. An SMD of minus 0.67. Still significant, but not as dramatic.
So we can deduce that if the depression is heavily linked to visible gut symptoms, a clear disruption of that MGB axis where the city wall is broken, FMT is more likely to be a powerful intervention.
That's the perfect interpretation. It highlights that FMT is most powerful for what we call disorders of gut-brain interaction. When the gut is visibly distressed, fixing the microbial community seems to have a stronger, cascading benefit that travels right up that superhighway to the central nervous system.
Let's discuss the delivery methods. Does it matter how we get the new community into the struggling city?
Oh, it absolutely matters, and the data was very clear on this. The method of delivery determined the level of efficacy and, crucially, the chemical changes that were achieved.
So what works best?
The clinical data confirmed that direct gastrointestinal administration delivering the microbes deep into the large intestine via colonoscopy or via enema was significantly more effective. This direct method had a greater effect size, an SMD of minus 1.29.
Which is even higher than the overall average.
Right, and that was much better than using oral capsules, which had an SMD of minus 1.06.
Why is there such a difference? I mean, capsules sound so much more patient-friendly and easier to scale up for widespread use.
They are easier, but they face a monumental obstacle: the highly corrosive, harsh environment of the stomach. Think of the capsules as little ships trying to deliver vital cargo, the new residents, to the city.
And the stomach is a sea of acid.
Exactly. The oral route exposes them to stomach acid, which acts like a powerful solvent, potentially killing a huge percentage of the new microbes and reducing their viability and functional stability. It's incredibly hard for the new residents to survive that treacherous journey.
But the direct delivery method, the colonoscopy or enema, bypasses that acidic river entirely and gets the new residents right where they need to set up shop in the lower digestive tract.
Precisely. And this was confirmed at the level of the brain chemistry, which is the mechanism we really care about. The group that received the endoscopic administration achieved superior modulation of neurochemicals.
What did they see?
They saw elevated levels of the calming, mood-boosting neurotransmitters serotonin and GABA, while the excitatory, anxiety-inducing neurotransmitter glutamate was dramatically reduced.
Wait, those are major brain chemicals. How long did it take for the bacteria to change the chemical landscape of the brain?
The change was relatively rapid, and the magnitude of these beneficial neurochemical shifts, calming the brain and boosting mood, far exceeded those seen with the less effective, less direct delivery methods like capsules. It strongly suggests that simply providing the right new microbial factory workers immediately changes the output of the chemical messengers used by the brain.
That is a stunning piece of information.
Yeah.
But you warned us earlier that this is not a permanent solution yet. What is a critical challenge regarding the long game?
That is the major hurdle for the field. While FMT showed significant, powerful benefits in the short to midterm, up to about 5 months, the overall positive effects tended to dramatically diminish by 6 months.
That's a massive problem. The effects wear off.
It is. It underscores the difficulty in achieving long-term stability and ensuring that the new microbes permanently colonize and stay functional in the recipient's gut. The old, established residents, the unhealthy ones, tend to be very resistant to change.
The new residents get pushed out.
Or starved out. The recipient's existing gut environment is often unfavorable to the new, healthy transplants. We are still learning how to make the new residents sign a long-term lease.
So we can restore order, but maintaining that order requires more than just a single intervention. We'll definitely have to dive into how to fix that stability problem later.
Okay, so if we move from the brain to the body, we look at the gut metabolism axis. Metabolic syndrome, METS, and obesity are arguably the greatest global health challenges of our time.
It's that cluster of conditions, right? High blood pressure, high blood sugar, excess belly fat.
That's it. And given that the gut is so clearly linked to inflammation and fat synthesis, many researchers banked on FMT as a major therapeutic breakthrough here.
Did the systematic reviews on metabolic markers yield the same kind of clear-cut success we saw, at least temporarily, with depression?
Unfortunately, no. The initial conclusions were, well, they were mixed, cautious, and statistically unstable.
Unstable, how so?
When researchers looked at the overall effect of FMT on the primary metabolic markers, changes in body mass index or BMI, insulin resistance, and long-term blood sugar control, they only found a trend toward improvement.
Let's define insulin resistance simply. They measure it with something called OMA-IR.
Right, OMA-IR. It's basically a calculation using your blood glucose and insulin levels that tells us how resistant your cells are to insulin. The higher the number, the more difficult it is for your body to manage blood sugar. It's a key marker of prediabetes and metabolic syndrome.
Okay, so what did the data show on these major markers like BMI and OMA-IR?
The improvements were statistically minor in the primary analysis. For example, the BMI improvements were small and didn't quite hit statistical significance. The p-value was 0.070, and we usually look for p-value less than 0.05.
So it was close, but no cigar.
Exactly. And the same was true for HOMA-IR. There was no significant effect on the long-term blood sugar marker, HbA1c, at all.
So if I'm the learner reviewing these sources, the initial headline is pretty disappointing. FMT doesn't significantly help with major weight loss or controlling blood sugar overall.
That was the initial conclusion of the raw primary analysis. But here is where we need to slow down and talk about the statistics because it reveals a massive real-world truth about FMT.
Which is?
The overall analysis showed a high degree of instability. The conclusion was extremely sensitive to methodological assumptions, meaning the data was highly variable.
Tell me more about that instability. What statistical assumption changed the outcome?
The researchers noted that simply adjusting one key statistical hypothesis, the correlation coefficient they used in their model, did make the BMI improvements statistically significant.
Whoa, hang on. A single tweak in how they handled the math flipped the conclusion from no effect to significant effect.
It did.
If the numbers are so fragile, what does that tell us about the real state of FMT for metabolism?
It tells us that we are dealing with tremendous variability in the real world. Think back to our city analogy. Some patients received transplants from a donor whose city was only mildly healthy, some from a super donor. Some patients got the transplant orally, some via deep colonoscopy. Some recipients had a very healthy starting diet, others had a terrible one.
So the statistical instability simply reflects the fact that some FMT attempts are massive successes and some are complete failures, and it all depends on these variables that aren't standardized. The overall average just washes out the successes.
Precisely. The field hasn't figured out who it works for, who the best donors are, and why it works when it does. We are still in the dark ages of standardization.
When speaking of success, there was a really compelling long-term bright spot in the data, particularly from a trial involving adolescents with severe obesity. This 4-year follow-up fundamentally reframes how we should view FMT success in metabolic health.
This study is crucial. It's so important because it showed that even if FMT doesn't cause the visible, dramatic outcome of immediate weight loss, it might be fighting the underlying silent damage caused by obesity.
Okay, so what did they find after 4 years?
After 4 years, the FMT recipients showed no statistically significant difference in overall BMI compared to the placebo group. The scale weight was the same.
But the underlying health of the city was vastly improved, right?
Absolutely.
Mm.
When they looked past the scale weight, they saw profound clinical benefits. The FMT recipients had a significantly smaller waist circumference, about 10 centimeters less.
10 centimeters, that's huge.
It's a massive indicator of reduced visceral fat, the dangerous fat around the organs. They also had nearly 5% less total body fat overall, and they had a reduced metabolic syndrome severity score.
So even without a change in BMI, they fundamentally restructured the way their body stored fat. They improved their body composition.
Yes. But the most dramatic finding and the most compelling evidence that FMT fights the disease and not just the symptom was the inflammation data.
The massive reduction in systemic inflammation.
It was huge. There was a 68% reduction in high-sensitivity C-reactive protein, HSCRP.
And HSCRP is that key marker of chronic silent inflammation, the persistent fire triggered by the leaky gut.
That's the one. This reduction signals that the FMT intervention, even if transient in some ways, successfully reduced the chronic fire that drives heart disease, stroke, and diabetes for years to come. They also had higher levels of good HDL cholesterol.
So even if the city didn't shrink in total population size, its internal systems, its public health, its safety departments, were vastly improved and less inflamed. That's a powerful distinction. How can scientists track these internal changes beyond simple blood tests? What are the specific molecular fingerprints of this post-FMT metabolism?
They look at the chemical changes in the blood, urine, and feces using advanced molecular profiling techniques. We can see profound shifts in metabolites, which are the complex chemical leftovers produced by the gut bacteria interacting with our food and our bodies.
Give us an example of a good chemical shift that FMT achieved.
One beneficial change was seen in amino acids. When patients received allogenic FMT, the healthy donor transplant, it was associated with beneficial changes. Specifically, we saw increases in GABA, the calming brain chemical we discussed earlier, and positive alterations in the tryptophan pathways, shunting more of it toward beneficial compounds.
So the new residents changed the internal factory settings to be less inflammatory and more calming. What about fat molecules, the lipids?
The pattern with lipids is particularly illustrative of the protective effect. The healthy donor group showed increases in beneficial lipids, such as 1-palmitoyl-glycerol. This specific compound is fascinating because it's known to activate brown fat, the energy-burning fat, and it aids in healthy insulin secretion from the pancreas.
So it's a hallmark of a healthy metabolism.
It is.
And what happens if the patient receives a transplant of their own feces, which serves as a control for the procedure?
This contrast is key. When patients received the autologous or self-transplant control, they often showed increases in detrimental lipids. These included compounds like sphingomyelin. These problematic lipids are strongly correlated with high insulin resistance and obesity.
So getting a healthy donor's microbiome actively pushes the recipient's metabolism away from these harmful pro-insulin resistance pathways and toward beneficial ones.
That's exactly what it implies.
Wait, let's talk about that moment where the bacteria truly seem to be taking over the command center. You mentioned the bacteria can change the host's genetic activity. That sounds like something out of high-end science fiction.
It sounds like it, but it's real science. It's called epigenetics. Epigenetics is the science of how external factors like diet or our microbial community can switch our genes on or off without actually changing the underlying DNA code itself.
So the DNA blueprint is the same, but the building codes on how to use it change.
Exactly. FMT caused considerable alteration in host DNA methylation. That's the molecular mechanism for turning genes on or off at specific sites in the responding patients. For instance, the methylation of the AFAP1 gene was significantly altered.
Why did that matter? What does the AFAP1 gene do?
The AFAP1 gene is involved in regulating cell structure and has been linked to mitochondrial function and inflammation. The profound implication is this. The microbial shift isn't just producing transient chemicals. The new microbial citizens are giving instructions, altering the building codes to the human host's genetic machinery.
They are acting as long-term modulators that can affect everything from how our cells respond to blood sugar to how much inflammation we generate. That level of control is breathtaking.
It is, and it solidifies the fact that the gut is not just a passive digestive tube, it's an active, powerful controller of our foundational biological processes.
We've established the deep links between the gut, the brain, and metabolism. But the sources show the microbiome's influence extends far wider than we ever imagined. Since the gut is fundamentally regulating the entire immune system, it seems logical that this would impact autoimmune and inflammatory conditions across every body system.
That's right. Because inflammation and a damaged gut barrier dysbiosis are at the core of so many chronic diseases, FMT research is quickly exploring these connections. Let's look at conditions where the immune system is misfiring.
What have they found regarding common inflammatory conditions, like skin diseases such as psoriasis?
Psoriasis is a great example. It's a condition where the skin cells grow too quickly, driven by an overactive immune response. Psoriasis patients frequently show reduced gut microbial diversity, their city is less populated and varied, and a specific imbalance. They have decreased beneficial bacteria and increased inflammatory bacteria.
So the skin inflammation is a symptom being driven or at least made worse by the gut imbalance.
Precisely. The hypothesis is that gut dysbiosis weakens the city wall, leading to systemic inflammation. This inflammation then travels and influences the specific T-cell responses in the skin, which are crucial to psoriasis. In preclinical mouse models, FMT from healthy donors successfully reduced skin inflammation and restored immune balance.
What about type 1 diabetes? That's an autoimmune condition where the body attacks its own insulin-producing cells. How is the gut involved in an attack on the pancreas?
Type 1 diabetes is a major area of research. T1D patients exhibit gut dysbiosis and, crucially, reduced levels of those key anti-inflammatory SCFAs we discussed.
Without enough SCFA fuel, the gut barrier weakens and inflammation rises.
You've got it. FMT is being studied here, often using an autologous approach, a self-transplant.
Why would they use a self-transplant for an autoimmune condition?
Because the goal is different. In T1D, they might collect stool from the patient when they are younger and healthier, before the autoimmune attack is fully established. They then reintroduce that healthy self-stool later on, hoping it can help preserve the patient's remaining insulin-producing cells by dampening the autoimmune response.
That's fascinating. Let's shift to the complex terrain of neurodevelopmental disorders like autism spectrum disorder, or ASD. Is the gut-brain axis active here too?
Very much so. Dysbiosis is strongly associated with ASD, and again, the leaky gut idea is central. This increased intestinal permeability is theorized to allow harmful metabolites to cross into the bloodstream and influence the central nervous system and brain development.
Are there specific bacterial bad actors in ASD?
Yes, there are often increases in species like Clostridium. These Clostridium species produce highly neurotoxic metabolites, most notably a compound called picresol. These compounds can get into the system and exacerbate ASD symptoms and contribute to neuroinflammation.
So we have specific bacteria producing a specific toxin that seems to be contributing to neuroinflammation and possibly behaviors in ASD.
Exactly. The gut is manufacturing chemicals that directly affect brain function and behavior.
And what about another complex psychiatric disorder, schizophrenia?
Schizophrenia patients also consistently show altered gut and even oral microbiomes, which are strongly linked to chronic low-grade inflammation. It all traces back to inflammation originating in the microbial systems.
All right, prepare yourself for the biggest surprise connection in the sources, the eye. How on earth do gut bacteria influence eye health, specifically a disease that damages the optic nerve, like glaucoma? This feels like the ultimate proof of systemic reach.
It is the perfect example. Glaucoma is a leading cause of blindness caused by damage to the optic nerve, and the connection is rooted in the same inflammation we've been discussing. Changes in the gut environment lead to systemic inflammation that can activate immune cells in the eye.
In the eye itself.
Yes, specialized immune cells called retinal microglia via pathways like TLR4.
Wait, so the same leaky gut inflammatory signals, the LPS molecules, that drive insulin resistance in the liver can travel all the way up, cross the blood-brain barrier, and activate the immune defense system in the back of the eye.
That is the hypothesis of the gut-eye axis. LPS escaping the leaky gut triggers that TLR4 alarm system, and those inflammatory signals then reach the retina and contribute to neuroinflammation there, which speeds up optic nerve damage.
Has that actually been tested?
It has, in fascinating preclinical studies. Mice receiving fecal samples from human glaucoma patients exhibited increased retinal damage compared to mice receiving healthy donor samples.
That is stunning evidence. It means that the composition of the gut microbiome, those trillions of residents, influences the severity of ocular disease through systemic mechanisms far, far away from the digestive tract.
We have established FMT's massive therapeutic potential across mental, metabolic, and autoimmune health. But now we have to address the most significant obstacle to making this a scalable treatment: the high inconsistency of results.
That statistical term you used earlier, heterogeneity, reaching up to 91% in some analyses. That signals massive variability. It means 9 out of 10 studies might look totally different. Why are the results so hit or miss?
It comes down to three major factors that are currently not standardized in research or clinical practice. These are the three weak links in the chain.
Okay, what's link number one?
First, donor selection is key. A successful transplant depends entirely on the health, robustness, and specific functional profiles of the donor's microbial city. We can't just use any random healthy person. Rigorous screening is crucial to exclude individuals with metabolic syndrome, diabetes, IBS, autoimmune diseases, and psychiatric conditions.
So you need more than healthy. You need optimal.
You need a super donor. The donor's specific metabolic characteristics, how well their microbes make scFAs and manage inflammation, are what drive the beneficial effects on the recipient.
So if the donor has a microbiome that's just okay, their transplant won't transfer those powerful benefits. That makes sense. What's the second factor?
The recipient baseline matters. Think of it this way. If you try to plant a thriving new garden in barren, rocky, compacted soil, it's not going to take hold. The donor microbial implantation, or engraftment, the successful establishment of the new strains, is strongly influenced by the recipient's starting point.
So what factors in the recipient matter most?
Their baseline microbial diversity, for one. If their current community is heavily entrenched and resistant to change, the new residents will be pushed out. Furthermore, their current diet matters immensely. If a recipient eats zero fiber, the new scFA-producing bacteria will starve before they can establish a permanent residence.
You're looking for a microbial match.
Essentially, yes. Higher compatibility between the donor and recipient microbiota predicts a better clinical response.
And the third factor circles back to what we saw with depression, how the transplant is administered.
Delivery route and regimen vary wildly across trials, from a single oral dose to multiple deep infusions over weeks. This variation severely influences outcome stability. We saw that single-dose FMT trials for metabolic conditions, for instance, often show that the beneficial changes return to the patient's original baseline status after just 12 to 18 weeks.
So it really is a short-term lease for the new residents. They clean up the city, but then they move out, and the old dysfunctional community returns and the problems resurface. The lack of sustained effect is the biggest hurdle to clinical adoption.
That is the pattern. The recipient's environment is resisting the colonization. This is why the stability issue remains central to the field. However, let's revisit the adolescent obesity study with the 4-year follow-up. That study gave us a crucial glimmer of hope for achieving true long-term change.
Right, because even though the overall microbial composition had mostly reverted and diverged after 4 years, something specific persisted.
Exactly. The study revealed that key donor-derived bacterial and, intriguingly, bacteriophage strains, which, remember, are the viruses that infect bacteria, actually persisted for 4 years.
4 years?
Yes. Even if the entire massive community shifted over time, these particular strains, or their viral partners, remained embedded.
That is the key insight for the future.
Yeah.
We don't need the entire massive ecosystem to take up permanent residence. We just need the specific, powerful, beneficial components, the super employees, to stay and keep producing the scFA fuel and anti-inflammatory signals.
That's the conceptual leap. It suggests that certain specific microbial or viral elements, rather than the entire massive community, might be the critical agents providing the lasting anti-inflammatory benefits seen 4 years later.
That makes the future goal clear. Stop transplanting the entire forest, which is variable and hard to maintain, and start identifying and transplanting only the specific, most valuable trees or seed packet.
That is the urgent move toward precision medicine in the microbiome field. First, we need to standardize protocols and donor selection. But the genuine future lies in moving beyond the broad, messy approach of FMT to highly targeted precision interventions.
What are those targeted future interventions?
We are looking at 3 main areas. First, engineered probiotics. designing microbes for a specific job, like making massive amounts of butyrate or consuming placresol. Second, using metabolite analogs.
So instead of transplanting the bacteria.
We can synthesize the healthy chemicals they produce, like specific SCFAs or beneficial lipids, and administer those directly, bypassing the need for the bacteria themselves.
And what about that fascinating discovery about the bacteriophages, the bacterioviruses?
Bacteriophage therapy is the ultimate precision intervention. Since the bacteriophage strains from the donor persisted for 4 years, targeting specific microbial viruses could be a powerful tool. We could use phages to selectively eliminate the harmful inflammatory-producing bacteria in the recipient's gut, thereby clearing the way for beneficial strains to establish stability and efficacy.
The goal in all these approaches is standardization and superior permanent stability.
That's it.
We've covered a truly remarkable amount of ground today, from the physical city walls of your intestines to the neurochemistry of your brain and the inflammation in your blood vessels. For the listener, the key lesson here is profound and cannot be overstated. Your gut is not separate from you. It is the core of your systemic health.
Absolutely. The gut microbiome is constantly dynamically communicating with your brain and body via a sophisticated, powerful system of chemical signals, inflammation pathways, and even genetic modulation.
In FMT, it offers significant therapeutic potential, especially for those gut-linked conditions like irritable bowel syndrome and the silent inflammation that drives metabolic syndrome.
But we have to be honest, the science is still young. It's wrestling with variability, stability, and the massive complexity of a trillion-resident ecosystem.
We're still learning the exact blueprints of the city, and which residents are crucial for long-term health. The field is exciting, but standardized cures are still on the horizon. Now for the final thought, something to contemplate as we wrap up this deep dive. Given that the success of FMT, the establishment of a healthy microbial city, is so heavily influenced by things we control, like our daily diet and the health of our baseline microbial diversity.
Is it possible that personalized dietary strategies, such as consistently consuming high-fiber foods that produce massive amounts of SCFAs, or adopting specific eating patterns like the Mediterranean diet, which are known to profoundly modulate the microbiome and improve both metabolic and mental health outcomes, are simply the most accessible and customizable form of autologous FMT we currently have?
That is a fascinating thought. By consciously choosing what we eat, we are essentially stabilizing and healing our own system without needing an external donor, simply by feeding the microbial city the right materials to thrive and fight inflammation.
Food is medicine, taken to the molecular level.
That gives us a lot to chew on. Thanks for coming along on this deep dive with us. Remember to follow us on your favorite podcast platform and see you on the next dive.